Hello students, we will continue the next topic of the chapter 5 in inheritance of one gene. Inheritance of one gene is like the monohybrid cross which you have studied in class 10th. So in that it's the first Mendel's first experiment, and what did he do in that? He cross pollinated tall pea plant with dwarf pea plant. So tall pea plant, capital T, capital T, you can see here tall pea plant and short dwarf pea plant. So in the F1 generation he got tall, all were tall plant. You can see here tall plants were obtained in the first generation. For F1 generation means first filial generation, filial means progeny. So all tall plants were in F1 generation. After that what did he did? He did self-pollination. He self-pollinated tall F1 plants. So F2 generation what he got? 1, 2, 3. 3 tall plants and 1 dwarf plant. So mostly they were tall and some dwarf plants were obtained. What was the conclusion of Mendel's experiment? He concluded that in F1 generation all tall plants were obtained, but dwarf trait was also present. So that is why in F2 generation 3 tall and 1 dwarf plant was obtained. So that means like you may be having some feature which your parents will not have, but your grandparents may be having. So that quality, that trait will be hidden in your parents generation, but will be coming out, or it will be expressed in your generation. So in the same way here also, all tall plants in F1 generation, all tall plants were there, but the dwarf character was there, it was hidden. In F2 generation that dwarf character was expressed. So in F2 generation 75% tall and 25% short plants were obtained. So with that he came to the conclusion that passing on characters from one generation to another is called inheritance factor, which is called gene. So the 2 copies of a trait are inherited in each sexually reproducing organism. Two copies of a trait means capital T or capital T small t. These are the 2 copies of a trait that are one you are getting from father, one you are getting from mother. Mendelian experiment, the phenotypic ratio that we get is 3:1. Phenotype means the outward appearance and genotypic ratio means the genes. So phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. So genotype means the genetic information. Example, capital T, small t, capital T, small t, small t. And phenotype means the outward appearance, like tall plant, the yellow color, whatever the outward appearance, that is called the phenotype. So organisms observed properties, that is tall, dwarf, round, wrinkle, they are called phenotype. Homozygous genotype means if the alleles of the same gene are identical, capital T, small t, small t. So they are homozygous. Heterozygous means alleles are different, capital T, small t, the alleles are different. Dominant and recessive trait. Dominant trait means the trait that gets expressed in the offspring. So these dominant trait takes over the other inherited trait. So they are the expressed trait. Example, free earlobe. Now in human beings you might have seen free earlobe. The earlobe will be having earlobe which is attached to the face. So that is joint, attached earlobe, and most of us will be having free earlobe. So this free earlobe is a dominant trait, and recessive trait is the trait that remains hidden and dominated by the it will be dominated by the dominant trait. It is remaining hidden. So the example is attached earlobe. So some of the dominant and recessive traits in pea plant is dominant round seed, recessive wrinkle seed. Dominant yellow seed, recessive green seed. Dominant is tall, recessive dwarf. Violet flower, dominant white flower, recessive. Inflated pod means the covering. Dominant and recessive constricted pod. Green pod is dominant, yellow pod is recessive. So when do the recessive traits get expressed? Recessive traits get expressed only in homozygous condition. That means when small t and small t are there, then only those recessive traits get expressed. So recessive traits that remain hidden, they remain dominated by the dominant trait that appears only if the offspring inherits a copy of the gene from both parents. So if from both the parents small t is being taken, then only that small t, that is the dwarf condition, will get expressed. So monohybrid cross means the cross between organisms that differ in one trait. So one trait, that is, here Mendel had taken only the height of the plant. So it is only one trait that is taken. That is why it's called monohybrid cross. Now we will come to what is test cross. Now test cross, in a test cross, an organism showing a dominant phenotype and whose genotype is to be determined. Suppose a tall plant. You don't know whether it is capital T or capital T small t. That you have to determine. So what is done? It is crossed with the recessive parent. So recessive will be what? Small t, small t. So capital T will be crossed with small t, small t. Capital T, small t will be crossed with small t, small t. That is a test cross. He is just testing and finding out what is the genotype. So the progenies of such a cross can easily be analyzed to predict the genotype of the test organism. So in that case, whatever progeny you get, with that we can identify whether it was capital T or capital T small t. So Mendel crossed the tall plant from F2 with the dwarf plant. Now you can see here one example of a test cross was given in which the dominant phenotype, this is the purple color flower. You don't know whether it is capital T or capital T small t, purple color. What is done? Test cross is done, crossing with recessive phenotype, that is small t, white flower. So you can see here, if PP is crossed with small p, that means this PP is crossed with small p. So you can see here P, small p. This is called the Punnett square. So P and P, they will form PP. P and P, this one, capital P small p, this one, capital P small p, this one. Here all the progenies that you obtain, they are heterozygous and they are purple color. Okay, now in this you can see capital P small p. This was the test cross that was done with small p. So here capital P small p purple, capital P small p purple, small p white, small p white. So this is how a test cross is done. So it can happen that it can when we are crossing, we will get white flower. So with that you can find out whether the this parent was a dominant what was the this one allele of the parent. Now look here, another example, homozygous recessive. Here the dominant phenotype, here look here, diagrammatic representation of a test cross. Now W and W, small w, violet and white flowers, we are considering that. So it can happen that this is a violet flower. It can happen that violet is represented by W, capital W, small w for white color. So it can happen that it can be capital W, capital W, or it can happen that it is capital W, small w. So what is then this capital W? It is test crossed with recessive. So that is small w. Here again it is test crossed with small w. So here what you get is, here you can see you get W, is it W, violet, W, violet, and here capital W, small w, violet, capital W, small w, violet, small w, white, small w, white. So when these progenies you get, you can you will be able to understand that the in the F1 generation the parent would have been heterozygous. When we get this, we can assume that in the F1 generation the parent was homozygous. Okay, so what is these boxes you see, they are called the Punnett square. It is a diagram that predict the outcome of a particular cross. It is named after Reginald Punnett. So capital Y, small y, it's YY, capital Y, small y, this it's written here, this and this it's written here, small y written here, small y written here. This is how you represent a Punnett square. See, similarly RR, it's written like this, RR like this, capital R, small r. So based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. So what are the two main laws which he gave, that was the law of dominance and law of segregation. We will see these in the next slide.